Welcome to the Structural Biology and Imaging Resources portal of the U.S. Department of Energy's Biological and Environmental Research. This video describes resources and capabilities in X-ray fluorescence imaging at DOE user facilities that are freely available to researchers pursuing BER science challenges. Hi, my name is Jocelyn Richardson, and I'm a staff scientist in the Structural Molecular Biology group at the Stanford Synchrotron Radiation Light Source, a DOE user facility sponsored by the Office of Basic Energy Sciences. At SSRL, we offer X-ray fluorescence imaging capabilities to scientists pursuing research in BER mission areas. XRF imaging is also available at a number of other user facilities, information on which can be found on the BER Structural Bio Portal website. X-ray fluorescence imaging provides information on the abundance and spatial distribution of an element or chemical species of interest within a sample. Most XRF imaging beamlines also have X-ray absorption spectroscopy capabilities, which probes an element's bonding properties. Information on the different types of X-ray absorption spectroscopy can be found in the X-ray absorption spectroscopy explainer video. Most commonly, XRF imaging is used to observe the abundance and spatial distribution of elements from phosphorus, although sometimes as low as magnesium depending on the abundance, through to heavy metals. XRF imaging beamlines perform X-ray absorption spectroscopy on a small spot on the sample surface. This differs to bulk X-ray absorption spectroscopy, which requires a homogenized sample and a much larger beam. At most XRF imaging beamlines, the X-ray absorption spectroscopy technique performed is XANES, or X-ray absorption near-edge structure spectroscopy. XANES spectroscopy provides information on the oxidation state or chemical species of an element. X-ray fluorescence imaging can go beyond mapping the distribution of total elements. When XRF and XANES spectroscopy are combined, maps of different chemical species can be generated. There are relatively few sample constraints to XRF imaging, which makes this technique amenable to complex and heterogeneous biological and environmental samples. At various XRF imaging beamlines across the DOE user facilities, samples of up to half a meter and down to a few hundreds of microns in size can be measured at the appropriate spatial resolution. Beam spot sizes range from 250 microns to 1 micron, and a range of X-ray energies can be applied. No other techniques can achieve such analysis with the complex and heterogeneous samples of BER researchers. XRF imaging can address a wide array of biological and environmental science questions. In recent years, XRF imaging has been used in the following types of BER studies: nutrient cycling in the rhizosphere with a particular focus on phosphorus, nutrient and metal uptake by model organisms in simulated soil environments, sulfur uptake and speciation by various microbial communities, the redox state of metals such as manganese and iron in the rhizosphere and how that redox state relates to microbial activity, the distribution and speciation of nutrients such as sulfur and metals such as arsenic and iron in various plant components such as seedlings, stems, roots, and leaves, and how the distribution and speciation of these nutrients or metals relates to a plant microbe symbiosis or external environmental pressures. Some other BER-related applications of XRF imaging include mineral microbe interactions, determining the speciation and distribution of sulfur and phosphorus in ancient marine environments, the stabilization of organic matter by minerals, and determining ecosystem health by looking at biomineralizing organisms. These are just some of the examples that are applicable to the BER community.